couple of slides are repeats of what we kind of talked about before we started the osmosis and diffusion. Just worded a couple of different ways, so I don't think I'll go back over the ATP powered and all that sort of stuff since we kind of covered that. So the other thing I want to get to for the plasma membrane, get to the slide that says endocytosis, because this is another way that cells can use their plasma membrane for getting materials. This is where if we take a cell and because the cell does have a plasma membrane, and if you remember when we started talking about it, and I forgot to get the video, but this plasma membrane, it can take little punches like this to it and just reform back up. But animal cells can, because we don't have a structured cell wall, animal cells can move in a fashion where if they come to or if there is a substance that they want to move in to themselves, they can do that. Because endo means to bring to the inside. So, being animal cells, we can get materials by a process known as phagocytosis. Phagocytosis, phago means eating, okay? So this would be like cell eating, all right? So if it comes across a particle, it can simply take that plasma membrane and begin to get to that particle and wrap around it. That plasma membrane, because it's a fluid membrane, can just simply form around the particle, and that particle is now inside the cell to be used. So they can have that type of means to get a material. They can also do something called pinocytosis, the drinking. So this would be cellular drinking. They can actually have this fluid to the outside of the cells, and the cells could form, once again, to just engulf some fluid. And whatever might be in that fluid will just simply move and come over to the other side of the cell. This is showing movement across the blood vessels, for example, where material, the cell just simply formed around it and moved it across and emptied it to the other side of the blood vessels. The other way that a cell can get material, it's termed receptor-mediated endocytosis. This means there's a receptor on that plasma membrane, and as the particles move move across that cell, when enough receptors have been activated, the plasma membrane simply folds in, and each of the receptors that had a particle are now inside the cell. So these are ways that being an animal cell, it can use the actual plasma membrane to get particles. And the goal with endocytosis, move the particles into the cell. If the cell can use the membrane to do that, to get materials, it can do that to get rid of the materials. When we have the cells of the body, okay, I mentioned quite a few times, every cell has a job to do. So if we think about it this way, once again, nucleus contains the DNA, which has the genes, which codes for protein. The only thing that can be made is the protein, okay? Now, some of those proteins, like we've seen with the plasma membrane, some of those proteins just simply need to go to the plasma membrane. Some of those proteins are used inside the cell itself. But then some of the proteins that get made are the product of that cell. Meaning, this is what that cell was supposed to make. For example, digestive enzymes by the pancreas. The pancreas is rather busy. We tend to associate it with insulin because of diabetes. But we also have the other job of the pancreas that does digestive enzymes to help us break down our foodstuffs. So, in order for it to get its product out of the cell, it has to move that product towards the plasma membrane and get it out of the cell. So exocytosis, exo meaning exit, to move out, okay? The cells of the body have to take their product and get it to the other side of the plasma membrane. Good examples, the digestive enzymes, mucus from salivary glands, secretion of milk by mammary glands, cells of the body. Whether we're talking a nerve cell, the cells produce a product, and the goal is to make sure they get their product to the outside of the cell. So now, up to this point, everything has been the plasma membrane. Everything. We now get to move to other parts of the cell. So that plasma membrane is busy, to say the least. So if we begin now to look at the other parts of the cell, for our purposes, like with our scopes, for example, the three things we should be able to see on a cell. We should be able to see the plasma membrane. We should be able to see the nucleus. We should be able to see the cytoplasm. Cytoplasm being the the inside fluid of the cell. So if we're looking at the cells, now that we've talked about that plasma membrane, okay, this fluid portion inside the cell, we're going to have it termed cytoplasm, but the area that we find dissolved molecules, which would be closer in a little bit, is cytosol. All right? So it'll almost look like inside is opaque, and closer to the outer portion would be more clear. So cytoplasm, cytosol. The fluid portion to the inside of the cell where we find everything. All right? It's giving an environment for the organelles to be existing in, for materials to be existing in, the cytoskeleton to be found, cytoplasmic inclusions like little vesicles, for example, and our organelles. So everything gets to kind of float around in this fluid part inside the cell. That's our cytoplasm. The cytoskeleton, just like the name implies, the cytoskeleton, it's a structure of, and guess what? Protein tubes, all right, these tubules that can exist, and they give structure to the animal cell. Like I said, we don't have a rigid cell wall. We just have this squishy membrane, but we needed to have some structure. Cytoskeletons are made up of some little tubules and some filaments that are present. Their structure that they create looks like a skeleton. And this allows for the animal cell to have structure, but at the same time, if the cell needs to, that cell can do this number, collapse in on itself. And then it can go back to its shape if it needs to. It's almost like allowing for the cell to be a pop-up tent that can be movable. The other part about this, because this cell has that little skeletal structure in it, it allows for those proteins that get made, they can like take a ride. So if you have a protein that was made on this side of the cell but needs to go to this side of the cell, it can simply take a ride on one of these filaments or these tubes. It's really cool. So it's just like its name says, it's for a skeletal structure. In some cases, all right, we see these tubes and filaments create projections. We can actually have that cell have a projection. A place where this is very important. In the cells of the intestines, for example, we get microvilli. When it does something like this, it increases surface area and it allows for more movement of materials. In the case where we might need it for movement, it will help create cilia, which are little hair-like projections off of the cell membrane. Think about your respiratory system, okay, where we've got these little hair-like structures, and it helps to keep anything that's in the air out here out. And if we do get it in too far, it helps us kind of begin coughing so that it doesn't get into our lungs. Okay, that's the best thing I can tell you about cilia. And then the sperm has the flagellum, okay, for the movement. Inclusions that might be found. Something such as a lipid droplet. That is a lipid droplet. When we consume food, the fats that get broken down will make a little droplet, and this is how we get them into the cell. That's an example of a fat droplet. So that would get into the cell would break the membrane down from around it, and those individual particles can be used by the cell. For example, making more phospholipids. When we talk about these organelles, that term simply means tiny organ.